Have you had your wisdom teeth pulled or know someone who has had them pulled? Why do so many individuals not have room in their jaw for these backmost molars to come in? Throughout evolutionary development, human jaw size has gotten smaller, leaving little room for these extra teeth to come in later in life. Even if wisdom teeth do come in, do we really need them? After all, our other molars are perfectly sufficient for chewing food. Wisdom teeth are an example of a vestigial structure, which lends evidence to evolution. Besides fossils discussed in the last part of this lecture, evidence for evolution is supported by anatomy, embryology, and molecular biology. We begin with comparative anatomy, which is the study of how body structures are the same or different among species. Comparative anatomy literally refers to comparing the anatomy among different species. One example of comparative anatomy is homologous structures. These are body parts that have the same evolutionary origin despite differences in current function or appearance. An example is the forelimbs of birds and mammals that have different outward appearances and different functions in flying, swimming, walking, or grasping items. However, the bones inside the forelimbs are the same, which suggests a common ancestor that changed over time. The similar internal bone structure of these appendages in the human arm, dog leg, bird wing, and whale flipper, despite differences in outward appearance and function, indicates that these organisms share a common ancestor. Another example of anatomy that lends support to evolution is vestigial structures. These are body parts that serve no purpose in the current organism. Examples include molar teeth in vampire bats and hind limb pelvic bones in whales and snakes. Vampire bats are called this because they suck blood, so they certainly don't need molar teeth to chew that blood. Hind limb and pelvic bones allow organisms to sit and stand upright and also to walk, which whales and snakes clearly do not do. Examples in humans include 3 ear muscles that we can no longer use, but help other mammals move their ears. Have you ever seen a dog or cat hear a sound coming from another direction and move their ears to redirect their ears towards that sound? What about wisdom teeth? We discussed these at the beginning of this presentation. Are they really necessary? Even the appendix, this is a tiny structure that hangs off of our large intestine and does serve a function in the immune response. But so many people have their appendices removed and are absolutely fine, so the argument is that we really don't need this organ anymore. So just why do organisms have these vestigial structures if they no longer use them? Well, ancestors had these structures and passed them down to their descendants. Throughout evolution, the human jaw has gotten smaller such that many individuals do not have room for their wisdom teeth to grow in behind the molars. These teeth become impacted in the gum tissue and must be surgically removed. Despite the fact that we are discussing anatomical features that come from a common ancestor, we have to keep in mind that there are many examples of anatomical similarities that do not actually stem from common ancestry. This is what convergent evolution is. These are similar adaptations that are produced in organisms that do not share the same evolutionary lineage. This leads to what's called analogous structures, which are outwardly similar, but they're non-homologous because they have different internal anatomy. An example is that both butterflies and bats have wings, but this similarity did not arise from evolutionary modification of a structure from a common ancestor. Butterflies and bats have similar outward appearances with similar functions in flight. However, the internal anatomy differs, so these organisms did not arise from a common ancestor. Another piece of evidence that lends support to evolution is embryology, which is the study of developing organisms before birth. All vertebrate embryos, which are those with a backbone, look similar early in development. For instance, fish, reptiles, birds, and humans all have tails and gill slits in early embryos. Fish are the only organisms that are born with both of these features. Reptiles, birds, and humans that reside on land certainly don't have gills, and humans certainly don't have tails. So why are these features present early on in development? That's because, once again, ancestor vertebrates had gills and tails since life originated in the water, and they passed these developmental genes on to their descendants. These are examples of homeotic genes that control structural development during embryonic development. All vertebrate embryos look similar early in development, including the presence of gill slits and tails. In humans, these are developmental genes inherited from ancestors that get turned off before developing into full features. Biochemistry, molecular biology, and genetics also provide evidence for evolution. For instance, cells and molecules from all organisms are very similar. One example of a molecule that's been conserved over evolutionary history is DNA. All organisms contain DNA as the genetic molecule that gets copied into RNA to make proteins. DNA has the same double helix structure in all organisms and contains the same bases in all organisms. This is because DNA as the genetic code molecule is very effective. Therefore, there's no need to change it as evolution occurs. Diverse organisms also contain similar genes. For example, organisms as different as bacteria share some similar genes with humans. So clearly, once again, our ancestors passed these genes down to us. Another example of a molecule that's conserved in all organisms is ATP. All organisms make and utilize ATP as energy. Proteins are another example of a molecule that's been conserved during evolutionary history. All organisms contain the same 20 amino acids that are coded for by codon triplets in messenger RNA. All organisms also use the organelle ribosomes to make proteins. Many proteins are similar in diverse organisms. For example, cytochrome C, which is part of the electron transport chain in mitochondria, is very similar in all organisms that have mitochondria. DNA can be used as a molecular clock to estimate the time when organisms diverged from a common ancestor. This is possible when biologists know the mutation rate for a gene and the number of differences in the DNA sequence for that gene in two species. For example, many human and chimpanzee genes differ in 4 to 6% of their nucleotides. Since substitutions occur at a rate of 1% every million years, it's estimated that 4 to 6 million years have passed since the two species diverged. Given the mutation rate of this gene and the number of differences in the DNA sequence for these genes, it's estimated that the two species containing these genes emerged from a common ancestor 50 million years ago.